Hello, this is class Bio 225, Genetics from Genes to Genomes, and this is Dr. Lucy Abgaryan. We are going to review Chapter 7, Part 1 today. For this chapter, we will talk about anatomy and function of genes, and we will talk about mutations. Mutations. So, what are mutations? What are gene mutations? Mutations are heritable changes in DNA base sequences. It is important to note that when there is mutation in DNA, it doesn't mean that the DNA itself is damaged. The molecule of DNA is not damaged. It stays complete. However, the genetic information is being changed. We can categorize mutations as forward mutation and reverse mutation. So the case of the forward mutation is where the changes happen from wild type allele to different allele. And the example is the albinism, for example. And the reverse mutation, which is more rare, is the mutation where the changes are happening so that the mutated allele goes back to its wild type sequence, wild type phenotype. And it is very obvious to assume that reverse mutations happen much rarer than forward mutations, because forward mutations can happen anywhere along the gene sequence. However, reverse mutations can happen only at the particular spot of the gene where the mutation already exists. Now let's talk about most common 4 types of mutations that exist. One is substitution, another is deletion, third one is insertion, and the fourth one is inversion. So now let's talk about all of this more in depth and try to understand what are the differences. So, substitution. Substitution is where the base of the DNA sequence is substituted or is replaced by another base. There are 2 types of substitutions. One is called transitional and another is transversional. So transition is where the same type of amino base is substituted by a similar base, like purine will be replaced by purine or pyrimidine will be replaced by pyrimidine. However, there is another type, which is transversion, where they will be replaced by the alternative type, where the purine will be replaced by pyrimidine or pyrimidine will be replaced by purine. There is another type of the mutation, the second one that we mentioned, the deletion, where one or even more base pairs are lost from our DNA sequence. The third type, insertion, is the mutation where one or more base pairs are being added to original DNA sequence. The fourth type, inversion, is the case when 180-degree rotation take place in DNA in particular segment. There is also a special case of mutations, which is the reciprocal translocation, where parts of two non-homologous chromosomes are changing their places. Now let's look at the visual picture of those mutation types. So the first one that we talked about was the substitution. And as you can see, the very first line, the very first sequence is our original sequence, and then a substitution with transition, where the same base pair type should be substituted with similar type. So here we see that we are having GC substituted by AT, and then there is a transversion where TA is substituted by AT. So where the purine is substituted for pyrimidine. And then the second case that we talked about, the deletion, and as you can see, the B sequence here is where we are missing entire fragment. The GCA fragment is completely missing, and the C insertion where we have additional fragment with two base pairs inserted between CG, and that's AA sequence that is being added there. So here is visualization of fourth type of mutation, inversion, and the first picture is the picture of the original sequence, where we have starting from the fourth nucleotide, it's CG CATGGTA, and it's going to, the mutation when it takes place, it's going to rotate it by 180 degrees, so we end up having TACCATGCG instead, which also by 100-degree rotation means that we are taking the strand of the complementary sequence from its 5 to 3 prime. So now it's becoming the initial strand 5 to 3. So basically that's what's happening here. And here is the visualization of reciprocal translocation. So we see chromosome 1 all in gray and chromosome 2 all in yellow. And we see that there are two fragments, right and left, for each one of them. And then after translocation, what we are seeing is that from two different chromosomes, one is ending up having left side from chromosome 1 and right side from chromosome 2, and the other one is having right side from chromosome 2 and left side from chromosome 1. So this is how reciprocal translocation looks like. As we explained, mutations happen spontaneously, and it's interesting to know what is the rate of spontaneous mutations in general. And there is a table with experimental data provided for rates of recessive forward mutations at 5 code color genes in mice, and it appears that 11 mutations are happening per gene for every 1 million gametes. And mutation rates in other organisms apparently are between 2 to 12 mutations per gene for every 1 million gametes for other organisms. And below is the table basically explaining how they got that number for 5 code color genes in mice. Mutation rates are different for different genes, and that's very reasonable to agree with that, right? Because bigger the genes are, larger the genes are, there are more mutation. There are chances higher to have mutations. So mutation rates can be between 10 minus 9 to 10 minus 3 per gene for each gamete, and the differences, as I already mentioned, are due to gene sizes, one, and also they are due to susceptibility of particular genes to various mutagenic mechanisms, for example, like UV radiation or other external factors that can cause gene modifications, gene mutations. Average mutation rate in gamete-producing eukaryotes is higher than that of prokaryotes, And the reason is that many cell divisions take place between zygote formation and meiosis in germ cells, and therefore there are more chances to accumulate mutations due to too many cell divisions. Here is a question for you. What do you think? Can diploid organisms tolerate more mutations than the haploid organisms? Please email me the answer to this question. So, do mutations in bacteria occur spontaneously? In 1943, Loria and Delbruck conducted an experiment to figure this out, and it was called fluctuation test. So what they did, they infected wild-type bacteria with phage, and of course what they have seen was that majority of cells was dying, but few cells could grow and still divide. So the question was, had the cells altered the biochemistry, biochemically were they altered? Did the cells carry heritable mutations for resistance? Or were these mutations arising by chance, or did they arise in response to the phage? So let's look at this experiment and try to figure those out. So for the experiment, Loria Delbruck had these two hypotheses. One is that resistance is a physiological response to a bactericide, and the second one was resistance is something that is arising from a random mutation. And then on graph B, on figure B, you can see that there are 11 different fluctuation test results, and it's not clear from these test results which one of these hypotheses is right. These test results are showing that any of these can potentially be right. So let's see how they address this and how they were able to figure this out. So they did this experiment that is with replica plating technique, where they have initial master plate that contains penicillin resistant and penicillin sensitive bacteria, and they grow it without any penicillin selection. And then they are doing this transfer where they are basically they are using this velvet technique where they are exposing this tissue to all of these clones, and then they are transferring it into new plate that has already penicillin medium in it. So if the genes don't have resistance to penicillin, they are not going to survive. And as you can see, only one clone that was from original plate with penicillin resistance is surviving and growing happily. In a graph, in a picture A, that's the bottom right plate. On very right side, you can see that one clone growing. And then, of course, there is a question, will it keep going? So is it some type of mutation that occurs and then is remaining within the gene? So in picture B, you can see that it shows that there is a master plate with many clones with penicillin sensitive bacteria. So again, the velvet technique is applied, and they transfer the contaminated tissue to the new plate, and then you can see first plate has only 4 clones in it, which are the penicillin resistant clones. The second one has, and the third plate has as well, which means that it is staying consistent. So it's occurring before the effect of the phage, and it is remaining with its gene sequence during all the next generations. Summarizing Loria and Delbruck's fluctuation test results, we can say that bacterial resistance is something that is arising from mutations that occur before exposure to bactericide. Bactericide becomes a selective agent. It kills non-resistant cells, and it is allowing survival of cells that have pre-existing resistance. Also, mutations occur as the result of random process. One such random event takes place, they usually remain stable. So let's see how some natural processes can change the information that is stored in DNA sequence. One of such processes is depurination, where the purine will be detached from the backbone of the DNA sequence. This process can be caused by high energy oxygen species. Therefore, like probably all of you are familiar with antioxidants. That's why people want to use antioxidants to not have this type of mutations happen within their DNA systems. And this takes place in every cell. The rate of taking this type of event is 1,000 per hour. It's pretty high, actually. Another natural process is deamination of cysteine. If you remember, cysteine is very similar to uracil. The difference was in amino group that was in that cysteine has as opposed to uracil. So during the deamination process, cysteine is going to change to uracil. And if you remember, uracil is the analog of thymine in DNA instead of RNA, and therefore we are going to have the mutation change where normal CG complementary pair will change to AT after the replication. Well, we all know that X-ray is a big source for cancer, and high energies X-ray can cause some significant damages in DNA. It can break the sugar phosphate backbone of DNA. And it can cause a deletion, which is one of the most unwanted type of mutations, because when you have a whole fragment deleted, it can cause shifting of the gene sequence so that it now will encode something completely different. Another type of effect or influence on DNA that we all are familiar with is the ultraviolet radiation light that is known to cause thymine dimers within the DNA backbone, and that is a covalent bond between two neighboring thymines, and it's important to know that it's not a mutation. It is a lesion in DNA. However, if it's not corrected, it will cause a mutation throughout the replication process. Besides thymine dimers, UV radiation can cause also a formation of free radicals that can alter individual bases. So we will have, instead of guanine, 8-oxo-DG, which is commonly called GO. And if it's not fixed within our sequence, instead of having AT complementary pair, we are going to have A GO complementary pair, which is not normal DNA nucleotide, and that will cause a mutation after the replication process. Note that ideally we have GC complementary pair, right? G is pairing with C. However, in this case, we are having A complementing with GO, which will cause AT mutation. And that's something that we don't want during our replication process. So now that we have talked about different factors that cause mutations or defects of DNA sequences, next time we will continue talking about how cells are implementing different processes to resist the mutations or to try to fix the acute damages.